you. I think it's been a great session so far, and I'd like to introduce her, our final speaker, who is Vanessa Wheeler. Vanessa is an associate professor at Harvard Medical School. She did her PhD at Imperial and then went over to the US and there developed and has sustained a very long-standing interest in the effects of DNA repair enzymes, particularly mismatch repair enzymes, on somatic expansion in Huntington's disease and other repeat disorders. And she's very interested in the mechanisms that underlie these phenomena. So I'd like to hand over to Vanessa now. Thank you.
I'd like to thank the organisers for the invitation and opportunity to present our data. These are my disclosures. In recent years, genome-wide association studies have provided significant insight into the pathogenesis of HD. The Manhattan plots on the left show genetic loci associated with HD onset in a GWAS of about 9,000 European individuals, with the most likely candidate gene at each locus indicated with an arrow. The GWAS provided support for a model in which somatic expansion of the uninterrupted CAG repeat beyond a threshold is needed to elicit cellular toxicity that drives onset. This threshold can be reached earlier with longer repeat tracks and can be modified in either direction by other factors. These data supported earlier studies in which we had examined CAG somatic expansion in individuals with extreme early or late onset, and in which we found longer somatic expansions in the brain were associated with an earlier age of onset. Quantification of somatic expansion across a wide range of central and peripheral tissues in HD patients revealed common tissue-specific patterns of instability across these individuals, as indicated by the heat map on the right. We also examined the instability of the ataxin-1 CAG repeat in SCAR1 patient brain tissues and found very similar patterns of tissue-specific instability to the HD CAG repeat, as indicated by the graph on the right. And these data indicated that the intrinsic propensity of the repeat to expand is independent of downstream pathology. Overall, the human genetic data support a two-step model for pathogenesis of HD that can be extended to other repeat expansion disorders that involves both a repeat expansion component and a toxicity component, such that pathogenesis depends on the rate of expansion in a particular cell type and the repeat length needed to elicit toxicity of a particular entity. So for example, although the SCAR1 and HD repeats might expand at similarly high rates in striatal neurons, a low repeat length may be needed to elicit toxicity in the context of mutant Huntington than in the context of mutant ataxin-1 in those cells. And this model implies that a full understanding of pathogenesis will need dissecting both the repeat expansion component and the toxicity component, with both providing targets for therapeutic intervention. My lab is particularly interested in repeat expansion, as this will provide proximal therapeutic targets that are likely to be applicable across diseases. To identify genetic modifiers of instability, we're using in vivo CRISPR-Cas9 genome editing. And here I'd like to particularly acknowledge Ricardo Moro-Pinto, who has spearheaded this work. Our approach is to use AAV8 to deliver guide RNAs targeting genes of interest to the liver of Q111 knock-in HD mice expressing Cas9. The liver exhibits high levels of instability, allowing a sensitive readout in a relatively short period of time. And we can quantify CAG expansion of the liver using gene map and fragment-based analysis of repeat-containing PCR products. To understand the sensitivity of this system to detect expansion modifiers, we carried out mixing experiments in which we mixed DNAs, stable and unstable DNAs in different proportions, and use these as substrates in the CAG expansion index assay. And this indicated that if we were to inject mice at 6 weeks of age and analyze them at 12 weeks of age, we'd be able to see an impact on instability if expansion was suppressed in 50% of the cells, or even as low as 25% of the cells. And using this paradigm of injecting at 6 weeks and analyzing at 12 weeks, we're able to achieve strong viral transduction in the liver and efficient gene editing, as indicated for a number of genes on the right. So we've used this system to test the impact of HD onset modifier candidate genes on CAG instability. And this table shows the genes that we've tested. Based on their biological function, a number of these genes are clearly implicated in repeat instability, whereas others are less clearly implicated. And a number of the DNA repair genes have already been shown to be important modifiers of CAG repeat instability in HD mouse models based on the impact of constitutional null mutations. The mismatch repair genes function as dimeric complexes, at least in the canonical mismatch repair pathway, as these mut-S and mut-L dimers, as shown in the panel on the right. This slide shows the results of testing these modifiers. The panel on the left shows the expansion index at 6, 12, and 24 weeks in untreated mice out of 12-week empty vector control treated mice. The middle panel shows the impact of targeting mismatch repair genes, and the right panel shows the impact of targeting other genes. Knockout of MSH2, MSH3, MLH1, PMS1, and MLH3 suppresses expansion to levels indistinguishable from those in 6-week untreated mice. Knockout of RM2B slightly suppresses expansions, whereas knockout of MSH6, PMS2, and FAN1 promotes expansions. And we were unable to detect an effect on CAG expansion by targeting any of the other genes. The table on the right shows the percentage of frameshift mutation in the liver, indicating that we are able to achieve sufficient levels of genome editing at these loci that would allow us to detect strong modifier effects. Overall, then, these data indicate that some of the HD onset modifier genes are likely to alter disease in patients at the level of CAG instability, whereas others may be more likely to act by other mechanisms, potentially altering the cellular toxicity component. However, it's important to note that this system provides one way of interrogating these genes, so that the lack of an effect really does not necessarily rule out a role in CAG expansion in disease modification in patients. For example, LIG1, the biological function of LIG1 clearly implicates this gene in repeat instability, and this needs deeper investigation. And conversely, an impact on expansion does not preclude other mechanisms of disease modification in patients. The role of FAN1 is of considerable interest in this process. FAN1 is not a canonical mismatch repair protein, yet has been found to interact with a number of mismatch repair proteins, including MLH1. To test the relationship between FAN1 and MLH1 in repeat instability, we carried out a genetic cross between Q111 knock-in mice, MLH1 null mice, and FAN1 null mice. Knockout, heterozygous or homozygous knockout of FAN1 promotes expansion in the liver and in the striatum. However, this effect is not seen in the MLH1 null, indicating that MLH1 is required for the destabilizing effect of FAN1's loss. We were interested to extend these genetic interaction studies to other modifier genes. And this slide shows the feasibility of doing this using CRISPR-Cas9 editing in the liver. where co-injection of 2 AAVs, each targeting a different gene, as shown on the right, does not result in an appreciable difference in genome editing at each loci compared to targeting each of those genes alone, as shown on the left. So we've used CRISPR-Cas9 editing to test some genetic interactions between modifier genes, as shown, and the combinations that we have tested are shown by the green squares on the right. We focused here on interactions between FAN1 and mismatch repair genes, and on interactions between mismatch repair genes that define the different mut-S dimers, MSH3 and MSH6, and the different mut-L dimers, PMS2, PMS1, and MLH3. And this slide shows the results of testing these genetic interactions. The panel on the left indicates the impact of targeting each of these genes alone, as shown previously. The next panel shows the impact of targeting FAN1 together with one of the mismatch repair genes whose knockout individually suppresses expansion. We're able to recapitulate the impact of the genetic interaction between MLH1 and FAN1 as shown with the constitutional knockouts, and additionally show that the ability of FAN1 knockout to enhance expansion is also dependent on each of these other mismatch repair genes. Similarly, the ability of MSH6 or PMS2 knockout to promote expansion is also dependent on each of these other mismatch repair genes. In the next panel, we targeted 2 mismatch repair genes, each of which individually, when knocked out, suppressed expansion. And because we didn't detect any novel phenotypes, we're unable to discern a genetic interaction between these factors. And in the last panel, we targeted pairs of genes that, when knocked out individually, promoted expansion and uncover some interesting genetic modifier effects. So knockout of MSH6 and FAN1 results in a phenotype intermediate between that of the MSH6 and FAN1 knockout. Knockout of PMS2 and FAN1 results in a phenotype that's indistinguishable from the FAN1 knockout alone. And knockout of MSH6 and PMS2, although each individually promoted expansion, doesn't produce any effect on expansion when targeted in combination. These data overall indicate multiple genetic and potentially quite complex interactions between these factors that indicate that they act in a common pathway to modulate instability. There are a number of considerations and implications of these data. First, because the mismatch repair components act in multiple dimeric complexes, it is possible that knockout of individual components may have indirect effects of altering the balance of the mut-S and mut-L dimers. Secondly, it's important to note that protein interactions or complexes that occur in the process of CAG instability may in fact differ from those defined by the canonical DNA repair pathways. And the involvement of FAN1 in this process is a key example of this. Thirdly, these are genetic studies that we have carried out, and combining genetic data and protein biochemical data will ultimately be needed to dissect mechanism. And finally, the interactions between onset modifier genes in the mouse that we've identified suggest that interactions between modifier alleles in humans are also likely. To summarize briefly some of the attributes and caveats of the liver CRISPR knockout system that we've been using. So this provides a relatively rapid test of candidate instability modifiers in vivo. We can benchmark well against known modifiers, is sensitive to detect strong modifier effects, and can be used to test genetic interactions. However, subtle modifier effects may be hard to capture. Modifiers may, of course, have tissue or cell type specific effects that are not captured in the liver. And knocking out genes does not capture effects of functional mutations, such as those altering specific activities or of the human modifier SNPs. Starting to address the last point here, we've explored the role of MLH3 endonuclease domain in CAG expansion. And to do this, we carried out a genetic cross between Q111 knock-in mice and MLH3DN mice. These mice, generated by Paula Cohen, carry a D1185N mutation in a conserved endonuclease domain of the MLH3 protein. And when we cross these mice with the Q111 mice, we find complete suppression at 3 months and 6 months of somatic expansion to levels that we see with an MLH3 null mouse. And this indicates that MLH3's endonuclease function is important for CAG expansion. So as a hypothetical model here for the roles of some of these proteins in CAG expansion, MSH2 dimers, which is the mut-S beta complex, may bind to certain repeat structures, recruiting a mut-L gamma endonuclease, MLH1, MLH3, that cleaves the DNA in a way that allows incorporation of repeats. This process is also dependent on PMS1, potentially as part of the mut-L beta dimeric complex as well. FAN1 intersects with this pathway in such a way to inhibit incorporation of repeats that doesn't allow expansion. There are, of course, many unanswered questions, and much of this is being addressed, beginning to be addressed by a number of labs. So what is the mechanism of FAN1 mediated suppression? How does FAN1 mediated suppression depend on these mismatch repair enhancing proteins such as MLH1, MSH3, and PMS1? What is the function of PMS1? And what I don't have on this slide is MSH6 and PMS2 that, when knocked out, promote expansion in the liver. What is their role? Is this due to direct or indirect effects, and what are their relationships with FAN1? In summary then, patient-based data support somatic expansion as the first component of the HD pathogenic process and a compelling proximal target for therapeutic intervention. In vivo CRISPR gene editing provides a powerful means to identify modifiers of CAG instability and their genetic interactions. And further functional dissection of modifier genes and pathways, in particular alleles from human GWAS, will be needed to understand mechanism and optimal therapeutic approaches. And in the last couple of slides, I wanted to touch briefly on somatic expansion as a therapeutic target, which will be expanded upon later in this meeting. And just to put up some key questions: what to target, what genes to target, how to target those genes, when to target, and how to assess efficacy. And so, touching on the last point, just coming back to our previous human data where we examined instability across brain and peripheral tissues, we find quite a good correlation between instability in the brain across many brain regions and in peripheral tissues. And this is somewhat encouraging because it indicates that in therapeutic trials, potentially peripheral tissues, peripheral readouts may act as a surrogate for instability in the brain. And with that, I'd like to acknowledge a number of people, the people who have directly contributed to the work I've described in this talk are underlined. But more generally, I'd like to thank the former and current members of my lab, my colleagues and collaborators at the Center for Genomic Medicine at MGH, and many other collaborators, and of course, our sources of funding. And I'd like to thank you for your attention.
Thank you very much, Vanessa. That was a fantastic, very interesting summary of all your experiments, looking at these, dissecting the pathways and really trying to understand the mechanisms. We don't have much time for questions. If you don't mind, I'm just going to give you one or two. I have a question from Katrine Bath at EHDN. Is there any difference in the mRNA level of FAN1 or MSH3 in human samples between controls and HD patients?
That's not really been looked at very well. So there are certainly some of the FAN1 and MSH3 human modifier haplotypes are associated with altered levels of those mRNAs. But in terms of HD compared to control, of course, that's just really in control samples, the EQTL studies. In terms of HD compared to control, that's not something that's really been looked at carefully.
Okay, thank you. And then there was a question from Leslie. Do you think that some of the effects on somatic expansion in mouse liver might result from the removal of the redundancy in the DNA repair systems when genes are knocked out, i.e. is the stoichiometry important?
Sure. Yeah, I think the stoichiometry could be really important. These proteins act in complexes and multiple complexes, and knocking out one could certainly alter the stoichiometry of the other remaining complexes. So that's something that would need to be looked at more. And, I think there's also potential for redundancy, that the lack of an effect when you knock something out. And other redundancies there, for example, Lig1 would be an example where you knock out Lig1, there may be redundancy with other ligases.
Thank you very much. I'm going to stop there if that's okay.
Okay, I'd just like to thank all of the speakers in this session for a great session. It's very clear that, the proximal target of the DNA and the things that act on the DNA are likely to be really important. And in fact, we will hear later in the conference from a number of people who are thinking about how to use these particular events as therapeutic targets in the disease. So I'd just like to thank all of the speakers once again, and Davina, my co-chair. Thank you.
Thank you, Leslie and Davina, and the speakers in that session. So we have a break now, and we'll be back promptly at 4:30 Central European time. So that's in just under 25 minutes. Thank you.